Welcome everybody to the Women's Global Health Institute Fall Symposium, which is focused on women's health in COVID-19 pandemic. I'm Dorothy Teagarden. I'm the director of the Institute, but I'm very pleased to introduce Dr. Marion Underwood, who is the Dean of the College of Health and Human Sciences, who will give opening remarks for our symposium. So welcome, Dean Underwood.
Thank you very much, Dr. Teagarden. Good morning. On behalf of the College of Health and Human Sciences, welcome to the Women's Global Health Institute Fall Research Symposium on Women's Health in the COVID-19 pandemic. We are delighted to have you with us and thankful for the technology that allows us to stay connected in these challenging times. Women's health has never mattered more and never been more worthy of research. Women's health must be studied in its own right because of sex-related biological differences, the fact that around the world women are of lower socioeconomic status and thus at greater risk for health problems than men, and also because health-related research has historically focused on men in ways that make women's health poorly understood. Some argue, and I agree, that racism is a public health crisis, but I believe it may also be possible to say that sexism is a public health crisis. The evidence is clear that the COVID-19 pandemic has resulted in greater burdens for women. Although it is becoming clear that COVID mortality rates are higher for men in most parts of the world, according to a recent article in The Lancet by Tali Berkey on the indirect impact of COVID-19 on women, women are more likely to bear the serious social and economic consequences of this pandemic. UN Secretary General Antonio Guterres noted that, quote, COVID-19 could reverse the limited progress that has been made on gender equality and women's rights, end quote. According to a UN policy brief published in April 2020, across the globe, women earn less, save less, hold less secure jobs, and are more likely to be employed in the informal sector. They have less access to social protection and are the majority of single-parent households. Their capacity to absorb economic shocks is therefore less than men. Also, according to Berkey, some 243 million women are thought to have experienced sexual or physical abuse at the hands of an intimate partner at some point over the last 12 months. Many of these women have been trapped with their abuser. Within 1 week of France instituting its lockdown, reports of domestic violence had surged by 30%. It was a similar story in dozens of countries, end quote. Around the globe, women have greater responsibility for the care of children and ill relatives during this pandemic, often at the expense of their own livelihoods, access to education, and health care. For all of these reasons, I applaud the decision of the Women's Global Health Institute to focus this research symposium on women's health in the COVID-19 pandemic. The College of Health and Human Sciences is so proud of the Women's Global Health Institute because we share several core values. HHS and WGHI value the importance of basic research, focusing on prevention and early detection of disease. We share a very strong value of interdisciplinary research, and we care deeply about improving health across the lifespan. The College of Health and Human Sciences is grateful for the support that the Women's Global Health Institute provides for female faculty, for faculty studying women's issues, and for our HHS faculty member, including a pilot grant to Xiang to study how estrogen receptor signaling might be a possible explanation for why inflammatory bowel disease may be more prevalent in women than men. We are also pleased and proud that one of our own HHS faculty members, Dr. Zhou Taylor in Human Development and Family Studies, will be giving one of the talks at today's symposium on challenges, well-being, and coping of single mothers during the COVID-19 pandemic. My congratulations to all on the excellent program planned for today's research seminar. Enjoy the day, and thank you again for attending, and to the Women's Global Health Institute for sponsoring this important event. Dr. Teagarden, I'll hand it back to you.
Thank you very much, Dean Underwood. I really appreciate your doing this for us. We greatly value and appreciate our partnership with the College of HHS and your support of the WGHI. It's very important. Thank you.
My great pleasure. Thank you.
So I am very pleased to go to our keynote speaker, Dr. Sabra Klein. Dr. Klein is a professor in the Department of Molecular Microbiology and Immunology at Johns Hopkins, where she also received her doctorate degree in behavioral neuroscience. She is a leading expert on sex differences in immune responses and susceptibility to infection, and currently has over 100 peer-reviewed publications, authored several books, book chapters, edited 2 books on the broad topics of sex differences in response to infection and treatments for infectious disease. During the 2009 influenza pandemic, she was commissioned by the WGHI to evaluate and publish a report on the impact of sex, gender, and pregnancy on the outcome of influenza virus infection. She is currently the president of the Organization for the Study of Sex Differences, which is the only scientific society dedicated to uncovering how males and females differ in health and disease, which we, of course, at the WGHI are very interested in. She's the PI of the Johns Hopkins Specialized Center for Research Excellence, or the SCORE, on sex and age differences in immunity to influenza. She's also received the Society for Women's Health Research Medtronic Award for Science Contributions, which recognizes not only her scientific contributions, but her mentoring other female scientists, which is also near and dear to our hearts. So I'm really very pleased to welcome Dr. Klein to virtual Purdue, and very much looking forward to her talk. Welcome, Dr. Klein.
Well, I just want to begin by thanking the organizers. I am thrilled to be here. It is just such an honor. I know the work of several in your Women's Health Institute, and I am just honored to be here to speak today about sex and gender differences in the pathogenesis of COVID-19. And so I would like to just start off by making sure that we are all in agreement on certain definitions. And this concept of sex as a biological variable, I think, has gained a lot of momentum, in part because of the NIH and the work of Janine Clayton at the Office of Research on Women's Health. And we define sex biologically based on our sex chromosome complement, our reproductive tissues or our gonads, and our sex hormone or sex steroid hormone concentrations. And so what I'm going to do today is point out that sex is a biological variable that affects immune responses, and I just want to point out some of the ways in which this can occur. So by sex chromosome complement, we are treating this as binary. Yes, there can be disorders of sexual development, and there can be genetic causes for this that that definitely occur, but in a minority of people. For a majority of us, sex is binary, unlike gender, which is definitely fluid. We either have 2 X chromosomes, which defines individuals as female, an X and a Y chromosome, which defines individuals as males. On the Y chromosome is the SRY gene, which encodes testis determining factor, which is going to cause this bipotential gonad to develop into a testis. And there are other hormonal factors that are associated with this. I'm definitely simplifying this function. In the absence of SRY, but we now also appreciate that there is additional signaling that is going on to cause this bipotential gonad to develop into ovaries that will secrete primarily after puberty high concentrations of estrogens as well as progesterone, not pictured here, the testes, both in utero, so there is an in utero surge, but then later, after puberty, will secrete higher concentrations of testosterone. We think a lot about this hormone-dependent signaling, the binding of these hormones to their respective receptors in the development of secondary sex characteristics, sex-specific responses in the context of reproductive behavior and physiology. But I want to point out that just about every immune cell in our bodies have receptors for sex steroids, and we know that sex steroids can transcriptionally regulate the activity of immune cells to result in sex differences in innate and adaptive immunity, the development of inflammation, and even autoimmunity. And I'm going to add to this list infectious diseases. In addition to hormonal dependent effects, sex chromosomes through X-linked genes can have direct effects on the immune system. And it turns out that the X chromosome is enriched for immune-related genes. And there is a growing body of literature from my lab and others to suggest that some of these immune-related X-linked genes escape X inactivation, meaning that then having these two X chromosomes can result in a dosage effect in the expression and activity of these X-linked genes. Genes in our autosome can also impact our immune system. Just last month, there was a beautiful paper published in Science showing that roughly 30% of the genes encoded in the autosome, so that's genetic material shared between the sexes, are sex differentially expressed. So I think as we continue to look for these sex differences, we will continue to find more ways by which sex could be impacting our immune responses. Much like the introductory remarks, I do not want to give the impression that biological sex is independent of what was referred to as gender constructs, or those social or cultural norms that may define our behaviors, our lifestyle choices, our nutritional habits, exercise, perceived stress, smoking, all of which can impact our perception of disease, our likelihood to seek out health care, our likelihood of using health care, decision-making, therapeutic responses, and even our occupations. So in the context of COVID-19, as it was brought up, roughly 70%, I think the actual number is 78% of healthcare workers worldwide are women. So in thinking about exposure. So these gender constructs, they're going to influence our behavioral patterns. They can result in epigenetic effects that can directly impact our biology, resulting in differences in the pathophysiology, manifestations of disease, as well as our responses to treatment. So I do believe that while we often attempt to study sex and gender separately, I think the concept of the intersection of sex and gender is a much more valid way for us to evaluate the epidemiology as well as the pathogenesis of infectious diseases. So sex differences, and you know, my focus is going to be on COVID-19, but I don't want to leave any of you with the impression that there is something so unique and different about COVID-19. What's unique and different about COVID-19 is that it is a pandemic occurring within our lifetime, and it has put sex differences more broadly on the radar of media and the lay press. But I do want to point out that there are sex differences that are reported in a number of diseases that are associated with our immune system. What I've done in this cartoon image is I've organized these diseases based on those that show the most profound female bias, moving over to those that kind of have mixed profiles, and then ending with those diseases that show a male bias. If we start over here, autoimmune diseases, so diseases in which our immune system attacks our own tissues, our own cells. So recognizing yourself is not something that your immune system should do. We have receptors on every cell in our body that our immune cells should use to know that this is not a cell or a tissue that should be attacked. It turns out that 80% of autoimmune disease patients are female. And some of those autoimmune diseases that show the most profound biases or female biases are listed. If we then go over to those diseases that show the most profound male bias, these include non-reproductive cancers. So Obviously, there can be a sexual dimorphism in a disease like cancer, where you only see this disease specifically in one sex or the other. Prostate cancer would be an example of that. Other cancers, such as breast cancer, can occur in both sexes, but to an overwhelmingly greater degree, in this case, in females as compared with males. So if we remove all cancers that occur within the reproductive tracts of males and females and look at what are referred to as non-reproductive cancers, we find that men are twice as likely to die from non-reproductive cancers as compared with females. Effects on the immune system and even the efficacy of immunotherapies are ongoing, and there are a number of groups, including at the Cleveland Clinic, who are studying these types of differences. Some of the cancers that show the greatest sex differences are listed here. I'm joining you from microbiology and immunology. I work with infectious diseases, which turns out to be rather difficult when studying sex differences, and I've dedicated the past over 20 years to this study. Some infectious diseases show a female bias, and those are listed here. Others of which that show a male bias are listed here. This was published in 2016, long before the SARS-CoV-2 epidemic. But I will point out that the closely related cousin of SARS-CoV-2, MERS, as well as the initial SARS-CoV do show male biases. So let's talk for a moment about this mixed bag of infectious diseases. So like I said, I've dedicated the past over 20 years to studying sex differences in infectious diseases, considering the mediators, and trying to better understand why some infectious diseases show a male bias and why others show a female bias. And I'm trying to summarize, an adult lifetime of research and analysis into a single cartoon image to meet the time requirements for my talk with you today. So humor me as I walk you through this cartoon image that a previous graduate student in my lab, Landon von Stieg, and I developed. What we're looking at are, on the x-axis, a continuum of the immune response going from a weak immune response all the way to a strong immune response. On the y-axis, we're looking at host damage. So this would just be the tissue damage, the damage that occurs as a result of being infected, which can range from a low level of damage, if we can control the infection rather well, to a high degree of damage, resulting in hospitalization and even death. So in microbiology, and specifically in infectious diseases research, we know that disease can show this, if you will, U-shaped curve. At one end, having a very weak immune response in which you cannot control the infection can lead to a lot of damage. And if we follow this curve around to the other end, we also know that some damage that occurs as a result of infectious diseases is not being caused by an inability to control the virus or and I'm going to just stick with virus because that's what's pictured here, but rather having an over-exuberant, possibly too strong of an immune response. Having all those immune cells and all of the proteins that these immune cells make enter into tissue is what can lead to damage and the development of disease. So what Landon and I did is we drew a stippled line down the middle, and we noted that there are definitely, and this is true probably for a majority of viruses for which sex differences either have been studied and reported, there is a male bias. And this male bias occurs in part because males tend to have weaker immune responses than females. And an inability to control these infections can contribute to the worst outcome of these particular diseases, including SARS-CoV-2. On the other end of the spectrum, There are definitely viruses, including influenza viruses, which I've spent a number of years working on, as well as HIV, in which females suffer a worse outcome. And we now know from work from my lab and others that it's the overexuberant, that very strong immune response and the inflammation associated with that contributes to tissue damage and worse outcomes. But our focus today is going to be SARS-CoV-2, and what I wanted to do in these introductory remarks was give you some perspective. So as was noted, females are more likely than males to survive SARS-CoV-2 infection. Many in the audience probably already follow this website, Global Health 5050. If you don't, make a note of it. Take a snapshot. It's a fantastic group, nonprofit, who are tracking sex disaggregated data from around the world. These are the most recent data that they have on their website as of the 2nd of November. Countries that have reported, and it should say sex disaggregated on cases and deaths, including in the past month, are in green. Countries that have not reported sex disaggregated data in either cases or death are in this kind of gold color, and those that have not reported any sex disaggregated data are in this orange or reddish color. And those that are not yet tracking, which there are only a few, are in gray. So you can see, I mean, we do still have a long ways to go. Not all countries are sex disaggregating data, and you can see this differs probably based on a lot of factors, including resources, finances, and you can even see on some continents there's diversity across different countries. The United States, there are differences across different states in terms of reporting sex disaggregated data. So the United States is not nearly as unified as it looks there. So the current picture from around the world for COVID-19 data is that for every 10 females, there are 11 cases in males. I know that makes it look, and I'm going to come back to this, as though there are equal numbers of cases for males and females. And while that's true, if we take all data from around the world, but I'm gonna make a point about some of the differences and discrepancies that we do see. Hospitalization, for every 10 females that are hospitalized, 12 males are hospitalized. For every 10 women, or females, I should say, that are admitted into intensive care units, 18 males are admitted into the ICU. For every 10 females that die, 14 males die. So this is from a piece that I published with several co-members of a working group, a gender and COVID-19 working group, which is an international group of investigators who are interested in sex and gender differences. And what I want to point out with you is that while it often does turn out that there are equal numbers of cases when we collect all sex disaggregated data from all countries, but what I do want to point out is that when we look at countries reporting sex disaggregated COVID-19 cases, we find that some countries are actually reporting a male bias, other countries are reporting a female bias, and still other countries are reporting no bias. And so while you may end up in the end with a roughly equal number of male and female cases, there are definitely differences. The pattern is not the same everywhere in every country. And I think that's very important, because when I see this, I think this speaks to many of the gender differences. In contrast, as was noted in the previous slide and in the introductory remarks, there's an overwhelming male bias being reported in the majority of countries around the world of COVID-19 deaths in men. So let's look at some data from France. These were data that were published roughly 2 months ago in Science. What I love about these data is, first of all, these were investigators, not invested, like many of us on this call, who are invested in sex and gender differences in women's health. So this was maybe a bit of a surprise to them. And here this was, sex-based differences being reported in Figure 1 in a science paper nonetheless. These were data that were collected from March through May, so during the first wave of the pandemic in France. The primary locations noted here in panel B, but what I want to bring your attention to are panel C, D, and E, in which they broke down their data, and you can see on the x-axis age groups, males blue, females red. And you can see that for both males and females, there is an increase in hospitalization, ICU admissions, and deaths with age. Okay, so females are significantly more likely to be hospitalized, to be admitted into the ICU, and to die at these later stages of life. But at every adult age examined, so from individuals from 20 years of age to over 80 years of age, there are significantly more men within each of these age ranges who are being hospitalized, admitted into the ICU, and who die. So we see these male biases not just in France, but we really see this across diverse countries and ages. So this is from a review that I did with Eileen Scully, who's an assistant professor at Johns Hopkins, as well as Kara Tannenbaum, who's the head of the Sex and Gender Institute at the Canadian Canadian Institute for Health Research, And what we've done is we've arranged countries based on their case fatality rates. So we have countries such as England that have reported, and this was during the first wave, a high case fatality rate, all the way to countries that were reporting a low case fatality rate. And what I hope you can see, turns out that 37 of these 38 countries reported a significant male bias. And you're seeing this male bias among countries that have high case fatality rates, again, such as England or the Netherlands, as well as in countries with low case fatality rates, including South Africa. Similar to what had been reported in France, there were 12 countries in our analysis that reported their case fatality rates broken down by age ranges. And so again, I think this intersection of sex and age becomes very important and provides us possibly with hypotheses about mechanisms mediating these differences. And again, what we saw was that across diverse countries, you saw the male bias in all adult age ranges, in this case ranging from 30 years of age to upward to 90 years and older. So how can sex impact COVID-19 pathogenesis? Let's start with the virus entering our cells. Respiratory epithelial cells are the primary cell type that are susceptible for SARS-CoV-2 infection. This is because respiratory epithelial cells express ACE2, which is the receptor used for virus entry. This is an X-linked gene. It's been shown in the kidney to be downregulated by estrogen. And it has been reported in the context of hypertension, both in humans and in animal models, that males express more ACE2 than do females. The fusion receptor, abbreviated TMPRSS2, is used to kind of anchor the virus. TMPRSS2 is androgen regulated in prostate cells, and data from the United States, as well as in Italy. I'm just quickly showing you some data from a study in Italy in which they looked at men with prostate cancer that were either on androgen deprivation therapy or who were not on androgen deprivation therapy. And they looked at whether these individuals were likely to be infected with SARS-CoV-2. And what they found was that men who were on androgen deprivation therapy were significantly less likely to become infected with SARS-CoV-2 as compared with men who were not on androgen deprivation therapy. This is correlation. This is not causation. And as you know, exposure to these viruses can involve many factors. But there are a growing number of studies across diverse countries, including in the United States, that are showing this. After the virus enters our cells, it must be sensed. And we have what are referred to as pattern recognition receptors that can sense the presence of something foreign like a virus in our body. SARS-CoV-2 is a single-stranded RNA virus, and single-stranded RNA is recognized by a pattern recognition receptor, abbreviated TLR7, toll-like receptor 7, which is X-linked, and my group and others have shown that this gene does escape X inactivation and is more highly expressed and active in cells from females as compared with males. So sex differences in the ability of virus to enter our susceptible cells, sex differences in the ability to sense the presence of the virus can lead to sex differences in the early innate immune response, which is going to be very important for initiating that immediate antiviral immune response within hours and days of infection, leading to sex differences in adaptive immune responses. So those are going to be the longer-term immune responses that are going to be necessary for clearance and long-term protection, memory, and hopefully protection against reinfection. I'm going to walk through some examples, including from my lab, for innate and adaptive immune responses, but ultimately, these differences in the immune system are going to lead to sex differences in the ability to control infection and resolve that inflammation, because remember, that disease, that COVID-19 disease, can be caused both by an inability to control the virus, but also having too much inflammation. And it's that sex differences, either in the control of infection or the resolution of infection, and or the progression of infection or the development of what's often being termed a cytokine storm, to reflect that over-exuberant immune response as though it's a storm in your body. So males, older adults, and patients with severe COVID-19 have greater cytokine responses. So cytokines are the signaling factors in our immune system. Several pro-inflammatory cytokines, including IL-6, IL-8, and tumor necrosis factor, or TNF-alpha, have been shown to be more highly produced in patients who are positive for SARS-CoV-2 than patients who are negative for SARS-CoV-2. When data are further analyzed, broken down by sex and age, we find that males are significantly more likely to have, or males do have, significantly greater concentrations of IL-6 than do females. We don't see that in this case for these other cytokines. We also find that older individuals are more likely to have higher concentrations of cytokines, not only IL-6, but IL-8 and TNF-alpha. If we come down here and we look at whether patients have low concentrations of cytokines or high concentrations of cytokine, and we model that against the probability of surviving COVID-19, individuals with lower cytokine concentrations, and we see this distinction to be the greatest for IL-6, individuals with lower concentrations are significantly more likely to survive COVID-19 than are patients with high concentrations of IL-6. We see similar patterns for these other pro-inflammatory cytokines, but not to the degree that we see for IL-6. Other data from a group at Yale have gone and looked at one aspect of the adaptive immune response, and that would be our T cells, specifically our CD8 positive T cells. So CD8 is just a receptor expressed on the surface of T cells that can define these T cells as being cytotoxic T cells, or T cells that have the capability of killing virally infected cells. Here they compared male and female healthcare workers, HCW, with male and female patients, abbreviated PT. It's important to note that the healthcare workers were an average age of 40 years of age, whereas the patients were an average age of 65 years of age. And I do think it's important because age, we know, is an important factor in the pathogenesis of this disease. When they looked at the frequency of all the cells that possessed the CD8 positive receptor, there was no effect of infection or sex difference in the effects of infection on the frequency or the numbers of these CD8 positive T cells. But when they looked at the activity of these T cells, so when they looked at a marker called CD38, which is important for the proliferation, the ability to be active, what they found was that female patients, so female COVID-19 patients, had more of these active CD8 T cells than male patients. When they looked at terminal differentiation, so the ability to completely differentiate and be a killer T cell, they also found that female patients, had more of these cells. Again, that intersection with age, when they looked at the expression of this marker of proliferation across age, or if they looked at cytokine production, so the production of a cytokine called interferon gamma by these CD8 positive T cells, what they observed across diverse ages ranging from less than 40 to over 90 years of age, among the males, so males being this orange color, this yellowish orange color, they saw a significant decrease in the activity of these T cells as measured by either proliferation or cytokine production. They saw a significant reduction with age in males. Okay, so that's this R value and P value listed here. For females, on the other hand, there was no effect of age. Females had greater activity, and this did not change with age, so that regression line was not significant, that association. So aging seems to have a larger effect in males than females on the activity of these cells. Well, my group has been very focused on the serological responses, and we have lots of different ways of measuring this, and I work with wonderful, wonderful groups of people. So this is by no means work by only my group, and I'll point out people during the acknowledgments. So if we look at immunoglobulin, so immunoglobulin G, which is the primary isotype of antibody that we have in circulation, we can look at responses to the spike protein, which is the protein that's going to bind that ACE2 receptor that I described to you. So we can look at a specific region called S1. We can look at the entire full-length spike, so the entire protein. What actually binds to ACE2 is called the receptor binding domain. So we can look at our ability to develop antibody responses against that specific domain of the spike protein. We can look at the function of antibody, the ability of antibody to actually bind to and neutralize virus and prevent virus from even being able to infect cells. And what we find when we use these 4 different measures is that the sex of the patient, the age of the patient, and the severity of COVID-19 are significant predictors of the antibody response. So let's go to a summary slide to make this a little easier to look at. So if we look at these adjusted coefficients in a logistic regression, we find that across these 4 different measures of antibody responses, males have greater antibody responses. Older individuals have greater antibody responses. People who are hospitalized have greater antibody responses. The only antibody response that was reduced over time in our population of 126 patients at Johns Hopkins was that neutralizing ability, that functional killing or neutralization of the virus by antibody. So when we talk about durability and we worry about reinfection, this could become very important. Well, one thing that we did, and really it was a wonderful graduate student in my lab, Janice Shapiro, who did this, we put all of the antibody responses into quartiles. For each of these different measures. So antibody that recognized the S1 of the spike protein, the full-length S, the receptor binding domain, or the antibodies that could neutralize the virus. And some individuals, so some of these 126 people, their responses to all of these measures fell into the lowest quartile. And those people got a score of a 0, and there were 16 people that fell into this category. Then at the other extreme, we had individuals that fell into the highest quartile for all of these responses. They received a score of a 12, and there were 17 people that met those criteria. And then you can see in the middle, we have every combination, with each row being an individual. So you can see there was definitely variability in the quartile in which their antibody response fell. When we analyze these scores in a logistic regression, and here we're looking at the adjusted coefficients, being male increased the score by 1.5 points. Being hospitalized increased the score by 5.2 points. And being older actually only increased the score by less than 1. And again, no real effect of time since a positive test. So being male and being hospitalized are predictors of greater antibody responses, which goes counter to what I've told you, that women tend to have greater immune responses than men. So this really went against our hypothesis, but we really are finding that individuals who have more severe disease have greater antibody responses. One interesting observation that was just published in Science last week is that males are more likely to have autoantibodies against interferon than females. So interferons, specifically type 1 interferons, are very important in that innate antiviral defense. And again, we should not be recognizing proteins that are being made by the cells of our own body, including our immune system. And what's been noted is that among individuals with what's referred to in this cartoon graphic with life-threatening SARS-CoV-2 infection are significantly more likely to have autoantibodies against type 1 interferons. Type 1 interferons being so important in that innate antiviral response, if antibodies, which are these Y-shaped factors in this cartoon, if the antibody binds up all of this type 1 interferon, the type 1 interferon cannot then protect these cells from infection. The virus can go in, it can replicate, and it can then be spread to additional cells. So we're not going to control the infection as well. It turns out that both sex and age are very important in predicting whether patients will have these autoantibodies. And it turns out that being male results in individuals being over 5 times more likely to have these autoantibodies. Being older increases the likelihood of having these autoantibodies by about 1.6 in terms of the odds ratio. They're 1.6 times more likely. So again, while the presence of these autoantibodies, they're low, so the percentage of people that have these are low, they are still significantly more likely to occur in male patients as compared with females, despite the fact that I told you that 80% of autoimmune disease patients are typically female. So you see, we're learning a lot. Males, as a result of these sex differences, presumably in the immune response, there's a lack of ability to control the virus in males. There are data suggesting that males shed more virus and for a longer duration of time than females. So the only published data to date in peer-reviewed journals has been out of Wuhan, China. So I'm hopeful that many groups, including our own group, so we're starting to look at this at Johns Hopkins to see if we're seeing something similar, including in the second wave of infection. I want to end with, again, not to ignore gender. Women are taking COVID-19 more seriously than men, meaning that women are more likely to be accepting of public health measures, including social distancing and mask wearing, than are men. And I want to point out that this cuts across diverse cultures and, in this case, countries. So, I mean, in terms of our public health messaging, trying to reach men to share the importance of public health measures, I think is very important. I work with a wonderful group, Hansel Park, Janice Shapiro, Becca Urson from my group did a lot of the antibody analyses. We did this with Andy Peckosh, Aaron Tobian, Arturo Casadevall, David Sullivan. I called out Eileen Scully. I am a member of the Gender and COVID-19 Working Group. And we just received I'm PI on a U54 from the National Institute on Cancer for a COVID-19 serology center. So with that, I thank you for your time. I will try to stop sharing.
Thank you very much, Dr. Klein. That was a really interesting presentation, and I'm particularly appreciative that you're able to explain the complicated immunology, which often eludes me, in a way that also includes depth, so I really appreciate that.
Thank you.
She would be willing to answer any questions that you may have, so please post questions in the question and answer box, and I will read them to her. Since we have so many participants, it's hard to not do it that way. But I will start off with a question. So it was interesting that androgen seems to intersect with this issue, but it would seem that estrogen does not play a role because you have no differences in age or postmenopausal women. Do you want to comment on that?
Absolutely. So, we do see an increase in severity of disease in postmenopausal women. Okay, so if you compare younger women to older women, just take the men out, you do see an increase in disease risk. So older age in women is absolutely a risk factor for more severe outcomes. But, and I think, to show you the complexity of this, even though postmenopausal women are at greater risk of more severe outcome than premenopausal women, they are still at a lower risk than their age-matched male counterparts. So, it's telling me that so in answer to your question, it tells me hormones can't explain everything. Hormones can't explain why a 90-year-old woman is significantly more likely to survive COVID-19 than a 90-year-old man. Because even at those kinds of ages, these are not people who are going to be on hormone replacement therapy.
Right.
That's not really getting monitored or discussed. So I don't have even answers about what hormone replacement therapy in postmenopausal women might be doing.
Interesting. Interesting. Okay, I have a question here from Carol Brophy. How do rheumatoid arthritis and asthma factor into prognosis?
Oh, that's so interesting. So how do rheumatoid arthritis and asthma? So I don't think, I think, and I have rheumatoid arthritis, so I've been following some of this, so I love that. And for everybody else in the room, rheumatoid arthritis is an autoimmune disease in which your immune system is attacking antigens in your joints. And so we are put on immunosuppressive medications. At this moment, I don't think the numbers of patients who have been studied have been adequate enough for us to draw conclusions. So we do have situations where some studies are suggesting increased risk, others are suggesting no increase in risk. So I think early on we were hearing a lot about asthma, but then as numbers of patients started to really increase in some countries, that significant effect of asthma went away. Rheumatoid arthritis hasn't been studied quite as much as asthma.
Interesting. Okay, well, I have another question from someone I think you know, Janice Evans.
Hi, Janice.
Go to Purdue virtually, Sabra. How many genes associated with COVID-19 infection escape inactivation? Are there more than TLR? And what is known about instances of Turner syndrome and COVID-19 risk?
Oh, I love it. So to answer the first part, we don't know. There is a group, so led by somebody who Janice likely knows, Priya Dougal, in the Department of Epidemiology at the Bloomberg School of Public Health at Johns Hopkins. So we are trying to look and see if we see any evidence of increased expression. I do want to point out, though, Janice, that there was a small cohort study, and it's a cohort study, and it was this interesting observation in 2 families where the males of the family were expressing mutations in TLR7. So you brought up TLR7. These mutations are not likely to present themselves in females because we are a mosaic of X-linked genes from our mother and our father. And having this mutation was associated in 2 unrelated families with much more severe COVID-19 in younger aged males. So these were men 20 to 30 years of age, one of whom even died. And it's very unusual for someone in their 20s and 30s to die. So I do still think TLR7 is important, but as Janice points out, it is not the only gene. It is not the only immune gene. It turns out we have like over 60 immune-related genes on our X chromosome, so I think the verdict's still out. The verdict is even still out whether we actually see escape from X inactivation in cells in the context of COVID-19. That's not been shown. Myself and others have done this in the context of other diseases. I've done this in the context of flu vaccine, and there are other groups who've done this in the context of HIV and autoimmune diseases. Her second question was, I can't remember.
And what is known about instances like Turner syndrome?
For Turner syndrome. So Turner's, if I recall, 2 X's and a Y. I don't know. I have not seen anybody report on that. And I think you can appreciate these are low numbers. That is partly why I just made the disclaimer that I was going to be a bit binary in my discussion today. But Janice, I will tell you that some nice work out of Oklahoma where they've got a really rich database of lupus patients have shown that Turner syndrome males are at greater risk of developing autoimmune diseases. They show a bit more of a female typic profile of autoimmune diseases. So, you know, if I had to predict, I might predict maybe they would show a bit, if there's something about the genetics of this, maybe I would hypothesize that Turner syndrome individuals might show a female typic pattern of susceptibility to COVID-19.
Okay. I have another question, which I think came from Richard Kuhn. Are there any gender differences in disease expression in children less than 18?
Such a good question. I don't know. One of the largest studies to date of kids with COVID-19, it's abbreviated BRAVE, so the BRAVE cohort, they didn't sex disaggregate their data. So I am working, you know, and I've made pediatricians aware that I would like for us to do that, to see if we see that difference. As you know, so in the paper from France that I cited in my talk today, and there's another beautiful paper from New York City during the first wave of the pandemic that I think was published in JAMA. They did not see sex differences in individuals under 18, but I also want to just, you know, time out. They also had very few cases in individuals under the age of 18, excuse me. So, you know, they're just so much more well-powered to then further disaggregate data by sex at adult ages because of the nature of the virus and the pathogenesis of the virus. It's adults who are more likely to suffer worse disease and be hospitalized.
Okay, interesting. So Susan South from Psychology asked the question of any thoughts on the increased blood clotting in COVID patients?
Oh, that's such a good question. No, I don't. I don't have anything profound to say about the blood clotting or the observation that still is on again, off again about our blood type. And I'm sorry, my dog is behind me having a good stretch. So I don't, but it is a good question. I've not seen anything, and if you have, please let me know, pertaining to sex differences in the context of that blood clotting.
So I have a couple more. So I have one from Belinda Suto, one of our external advisory committee members, who addressed social determinants of health and how stress hormone plays in COVID-19.
Oh, that's so important. You know, there really hasn't been a lot. I've not seen anything where anybody has looked to measure glucocorticoids in patients with COVID-19, but as you know, that is the steroid that is being given and is working better when administered to patients later in their disease, presumably because it is dampening the inflammatory immune response. But in thinking about the social determinants of health and thinking about how stress, either through being a caretaker, being a healthcare provider, being an essential worker during the early stages of the pandemic and having greater risk of exposure, I think all of that is very important, and I would predict that in that context of thinking about some of those social determinants, again, like the introductory remarks, I would think it would be women who would be more affected by this and by that stress.
Yeah, I think there's some evidence out there to support that. I'm not as familiar with it, but I have another one from Harm Hoganish, which not surprisingly is from the vet school. Can you comment on sex differences in animal models of SARS-CoV-2 infections?
Thank you. And this question was not planted. I love that you asked this. So animal models for SARS-CoV-2 is a really hot topic because most of us, myself included, the minute, there's an infectious disease outbreak out there, we want to stick it into mice. Why do we want to stick it into mice? Because mice are wonderful. We have every genetic mutation. We have every reagent you could ever imagine to study whatever we would like in the context of infectious diseases in mice. Turns out mice are not susceptible. So myself and others at Johns Hopkins are working in a hamster model. Turns out you can give hamsters clinical isolates from people, clinical isolates of SARS-CoV-2 from people, and they get sick. And we are seeing a sex difference. We are finding that males are suffering more, so they're getting sicker. Inflammation in their lungs is worse. We have some preliminary evidence suggesting that viral titers in the lungs might be higher, but these are low sample sizes because we're running this as a series of replications. We're also finding, and this is over 3 replications, that in the hamsters, the antibody response is actually greater in females than in males. So, it's not to say these models are going to be perfect or going to mirror everything we're seeing in people, but at least we are seeing that the outcome of infection is worse in male hamsters than in female hamsters. So stay tuned for that.
That's so important for our ability to be able to really study this well. So that's great news.
Thank you.
We will continue here. So from Debbie Mix, is there any evidence of gender difference regarding length or after effects healing time of infection patients?
I love that question. So yes. So there are data, I want to say they're out of Spain, showing that women are significantly more likely to be in that, what are now being referred to as the long haulers. So even though we may be less likely to have as severe of disease as our male counterparts, we are more likely to have some of those long-term effects, the fatigue, the cognitive fog, and I'm sure everybody on this call can appreciate this. when you have these nondescript, non-specific, long-term symptoms of an infection that has been cleared, and it's women, you know, you run into some of the biases that occur of saying, well, maybe it's just the stress of being in the pandemic, sending women for psychiatric evaluation and not necessarily taking these symptoms as seriously as they need to be taken. We see something very similar with Lyme disease. So, you know, they haven't necessarily been called the long haulers, but that long-term Lyme syndrome of fatigue is significantly more likely to be seen in women as compared with men. So we're now at Hopkins tracking, and we are seeing that women are more likely to report symptoms for a longer duration of time. So we haven't yet gotten to where these are the long haulers, but they are, you know, more like, you know, and again, is it sex, is it gender? Are women just more likely to, is it a reporting bias? Are we more in touch with how we feel? You know, I mean, so you can see the complications of interpretation, but great question, and yes, women are more likely to be reported as long haulers.
Okay, right now I have one more question from Suad Amiyar. Hope I pronounced that correctly. Hi, thanks to Dr. Klein for this great talk. Can she tell us more about the pregnancy and if there's a difference in the response to COVID in pregnant women versus non-pregnant women of the same age?
Oh my goodness. Okay, so, and this wasn't a question that was planted. We just submitted our paper. I work closely with an OBGYN at Hopkins, a dear friend and colleague, Irina Bird, who I love and is somebody that you guys should probably have come and speak about these issues. So it does turn out that there are some differences associated with the immune response during pregnancy. And what we're finding is that women that have, pregnant women have a very robust inflammatory response within the first 14 days after confirmed infection. So, you know, those inflammatory types of immune responses that I talked about among non-pregnant individuals that can be associated with more severe outcome, pregnant women are more likely to show that pattern very early. We're also finding that they have lower antibody responses, and they are significantly less likely to produce those detectable neutralizing antibodies, those antibodies that are going to kind of bind up and neutralize the virus. They're less likely to have that type of antibody than age-matched infected non-pregnant women. So we do think that could be important. These are observations, so I can't, you know, it's correlation. These are just, you know, convenient samples. These were women who came to Hopkins, were confirmed to be positive, delivered at Hopkins, and we were able. But we are even seeing some of those inflammatory immune responses on the fetal side of the placenta, which isn't good. You don't want inflammation in the placenta, and so we're reporting this.
Very interesting. I think we're more as our populations grow and we get more information.
Absolutely.
So I'm just glad to hear that the research is being done and the disaggregation, some, and hopefully there will be more of that information available.
So too.
So thank you so much, Dr. Klein, for really interesting and willing to answer all of our questions.
I have loved this, and I plan on staying with you today. So thank you.
So this will end our morning session, but please keep in mind our pilot grant submissions, which if you're interested in learning more about the Institute or that, you can check out our website. And this recording of this symposium will also be available on the website. So we're going to take a break, so grab your lunch, maybe some more coffee and tea, and come back and join us at 12:30 for the second set of our really interesting talks. So again, thank you, Dr. Klein.
Okay, I think it is 12:30, and I welcome you all back to the afternoon session of the workshop on women's health in the COVID-19 pandemic. I'm Ulrike Deidag, the Associate Director of the Women's Global Health Institute, and I have the pleasure to moderate the next session. We now will have 4 speakers from Purdue and IU sharing their research on this subject. And just as a reminder, each of the talks will be 10 minutes long with some 5 minutes time for discussions. So to the speakers, I will switch off my video and I will switch it back on about 1 minute before the 10 minutes are over, just as a little sign that the talk should come to a conclusion. And to all of you attendees, please feel free to already start writing your questions into the Q&A box already during the talks. We will be moderating them at the end of each talk. And with that, I'm very happy to introduce our first speaker, who is Dr. Craig Goergen, who is the Leslie A. Geddes Associate Professor of Biomedical Engineering at Purdue University. He's also the Director of Clinical Programs in the Wellbeing School of Biomedical Engineering and Adjunct Associate Professor of Surgery at Indiana University School of Medicine. He will be talking about early detection of COVID-19 by monitoring resting heart rate and respiration rate using wearable devices.
Thank you, Dr. Dida. Can you hear me okay? Wonderful.
Yes.
Wonderful. All right, so I'll go ahead and get into presenter mode. Hopefully everybody can see. Thank you so much for the opportunity to present today and tell you a little bit more about our work in this space. Again, we're focused on a project with a wearable device to early detect COVID-19 through biometric data. Briefly though, my lab really is focused on cardiovascular disease in general. We do a lot in animal models looking at heart attacks and aneurysms with strain assessments. We develop new technologies with photoacoustic imaging for compositional information. We're thinking about device development, collaborating with others in the department here to think about ways to look at the heart, how it changes after a heart attack, all related to cardiovascular disease. This work then has led to some wearable device work, including some funded by the WGHI at Purdue on preeclampsia development. We've got interest in see if we can develop a wearable device and a wearable strategy to not only diagnose preeclampsia, but detect and predict preeclampsia development before it happens. And so with that, we were making great progress. Everything was going well, and then spring 2020 came around and COVID happened. And so our lab shut down. Students had limited access to the building, if any at all. Anything collaborative or in teams was basically shut down. And I don't know if anybody else felt this way, but we were pretty helpless. I didn't know what to do and how to contribute. So we were realizing, this isn't going to be a 2-week issue, right? Even now, our cases are rising. We have a positivity rate on campus over 5%. We're going to be thinking about what we're to do in the near term, but also in the long term, and that even with vaccine development, we're going to be having issues over the winter months as well as beyond. So with that, and with the wearable strategy, we started looking into the literature, and we're realizing that there's something to think about with these biometric wearable approaches. And so there's been a few studies now that have come out, and one of the largest one has been with Fitbit. And this is a retrospective study. It's just a preprint that came out really recently, but with over tens of thousands of subjects, they went back and identified over 1,000 that had a confirmed positive COVID diagnosis, and then looked back retrospectively at all their Fitbit data. So based on their baseline, each of the individual patients, heart rate starts to rise, respiration rate starts to rise, even before symptoms set in. The RMSSD, root mean squared standard deviation of the heart rate, or basically how much heart rate variability there is, decreases before symptoms set in. And with that, we're thinking maybe there's something we can mine here, something we can be useful to know. And so we then connected with this really innovative, great company based out of Chicago. Gary Conkright is the CEO of PhysIQ. He's also a Purdue alum. And their strategy is to build analytic platforms using biometric data from a variety of third-party wearable devices. His team includes several other Purdue alums. Mackenzie Tuerty is from the BME department as a research engineer. Stefan Wegerich is the CSO. Karen Laramere is the director of clinical development, and Yorma Vaughn is operations project manager. And they've got a ton of experience working with health systems, hospitals, companies on taking this biometric data and building AI platforms that can then use it and make clinical decisions from that. And so Purdue and the Research Foundations actually invested in them. Even just back this past January, to advance these AI innovations with a $500,000 investment from PRF. So in conversations then with others on campus, including George Wadika, head of BME here, Ed Delp, and Maggie Zhou in Purdue Electrical and Computer Engineering, we all have interest in this wearable space, background in signal processing, device development, and wearables. we started partnering and thinking about what to do. And then the Executive Vice President for Research at our office put out this Protect Purdue Innovations Faculty Grants program. And so like a lot of things now, speed is of the essence. So this was announced in June. We put something together in July, and we got the funding in August to start a project to partner with PhysIQ and actually develop a feasibility study. So what we were doing and thinking about is using commercially available products. In one case, the Samsung Galaxy Active 2 smartwatch. I won't get into all the engineering details, but it sends out light through a light-emitting diode, which then bounces off in the capillary vessels red blood cells that then can be detected by a photodetector. And with that, we can get this PPG signal, photoplethysmography. And with that, you can get heart rate just based on the cardiac signal, and then filtering out respiration rate based on this respiratory sinus arrhythmia. So your heart rate actually changes as you breathe in versus breathing out. One quick note on sex differences here is that the watch band that comes with the Samsung Galaxy Active 2 is great for the majority of people, but it was tested on men, I'm pretty sure, because the vast majority of the female grad students and others that have tried this out that have smaller wrists, it was not even close to fitting. So what we realized pretty quickly is that we needed to provide a couple of different sizes of wristbands, because it's really important with this device to have a close, tight watch fit so that the watch sits nicely on the wrist to get good signal. We wanted then to compare it to what we considered a gold standard. And so while the wrist is a great place for long-term monitoring, we weren't sure about the quality of the data. So we ended up using a Connect Vital patch, which can be placed on the chest and with measuring electrical activity directly, as well as the stretch on those two sensors and a battery that we can detect respiration rate over a 5-day period with this Vital patch. And then in combination then with the smartphone, this PhysIQ app is great for connecting to the patch, and then while the subject uses a watch and a patch at the same time, the watch actually sends the data through cell signal to our cloud-based system. We can monitor ECG, respiration rate, and movement in general. So our feasibility study came together with something like this. We were going to identify and target the Purdue community, staff, faculty, students, and people around in the West Lafayette area, to monitor over a 5-day period, show that we can actually have people use these devices, whether it's the patch or the watch, with a closed feedback loop system, then send data to a cloud-based platform, and then using these algorithms, look at individual subjects and get insight on what's baseline for them and how things are changing over that period. So with continuous physiological monitoring via wearable sensor data study, we came up with the acronym Common Sense. So right now we have approval from the IRB for a 100-subject study. The aim one is to just collect biometric data from the Purdue community using these wearable devices, both the Samsung Galaxy Active 2 watch as well as the Vital patch. And then we're working on analyzing the data. And we're just in initial stages on this aim, but looking at the biometric signal to monitor movement, heart rate variability, and respiration rate. So far, we've got 55 subjects participating. 33 have been completed the 5-day study. 22 are either pre-screened or scheduled to begin in the next few weeks. We're currently enrolling anybody 30 years and older, so if you're interested, please do reach out. We have a lot of students in that 18 to 29 range that have already participated, but we're looking for people in older age ranges to get a larger range of subjects. Our group is focused more primarily on conducting interest employments and acquiring information for informed consent. The patient support is from us, as well as the incredible PhysIQ support team and patients support team. And then data analysis is both our group and then Maggie Zhou's group and Ed Delp's group and ECE to develop and work on these advanced algorithms. So a couple of just quick examples. The good PPG signal looks something like this, where we can detect with filtering both respiration and heart rate. Bad PPG signal, whether it's motion artifacts or other problems, whether a fit of the watch looks something like this. But even then, with some more sophisticated approaches, we can pull out respiration and heart rate based on that. So looking at sex differences, we did go into the data a little bit, and this is not only ours, but also previous data that PhysIQ collected. But looking at pink is female, blue is male, under 30, 30 to 50, and 50 and above, we found that the resting heart rate is going to be slightly higher in women of all ages. the resting heart rate variability, how much it changes normally at rest, is slightly lower in women of all ages. And resting respiration rate is slightly higher in women under 30 and 30 to 50, slightly lower, but about the same in women 50 and above. So this is important, especially the heart rate variability, as we're starting to identify what is baseline and normal, and then thinking about what would be a flag. What would tell somebody, hey, there might be something happening. You should go think about getting tested because there's some early signs of viral-like infection. So we're going to use these data to help think about and develop novel machine learning algorithms that can process this large amount of continuously generated biometric data, really with a focus on identifying early signs of decompensation where heart rate is increasing, resting heart rate is increasing, even though the subject is not moving around or exercising. And then we're also going to think about sex differences as well. Really thinking about what do we want to use as for algorithms of men versus women, and how are we going to use sex as a variable. Eventually, I think the next step is to conduct a larger study, include additional students, staff, faculty, others in the Purdue community that could be considered high risk, or even those that have had a confirmed positive test, all with the idea of eventually creating a full-scale monitoring program available to Purdue to use biometric data if they want for early possible detection of COVID-19 infection. Obviously, then you need to go and confirm the diagnosis with a test of some kind, but biometric data we think can give us an early clue as to what is happening for each subject. So with that, I need to thank the labs, including Jen Anderson, who's done a fantastic job coordinating the study, as well as Noah Nye, who's helped out with a lot of the patient consenting so far. Maggie Zhou, Ed Delp, and George Wadika, collaborators at Purdue, and the incredible team at PhysIQ, Karen, Mac, Yorma, Stefan, and Gary, thank you so much for your contributions and help on this and getting Purdue open again. Funding as well from the Purdue Innovations Faculty Grant, as well as the Women's Global Health Institute. And with that, I'm happy to take any questions.
Thank you so much for this wonderful talk. That is really interesting. So I encourage everybody to please enter your questions in the Q&A box. In the meantime, I have a question. So if I understand correctly, Craig, are you still staying?
Yes, I'm here. I don't know why I went off. Okay.
It's all based on heart rate and the change of heart rate at a particular time. So how specific would this methodology be? Because, I mean, heart rate changes due to all kind of other reasons as well.
Yeah, and that's why we're key on putting the word resting heart rate. And so if you go for a run, if you start walking up and down stairs, your heart rate's obviously going to change, but we're focusing on periods of rest. And so that's where that motion information and activity of the wrist is going to help give us a sense of what the subject is doing normally.
I was actually meaning also about resting heart rate, not when you run. But so basically assessing the variability now, that's kind of your idea of then you need to be way above that, and your preliminary data suggests this from the data. Okay, great.
Yeah, and the patch might give us more high quality data than the wrist, but we're going to use that to really do that comparison. And then some people will have higher heart rate variability than others, but we're going to use that as a baseline metric then.
Okay, wonderful. Are there any questions from the audience? I do not see any at this point yet. Are you thinking of commercializing this technology if it works out?
Yeah, the great thing that PhysIQ has the AI platform already FDA approved and are thinking about ways to expand it for a wide variety of uses. And so, we're really thinking to partner with them, and there are some ways to think about more advanced algorithms that might be interesting for licensing, but I think PhysIQ has the interest in commercialization and taking it to a broader audience.
Okay, well then, thank you so much again.
Great.
And I will continue. Next, I'm pleased to introduce Dr. Gregory Sokol, Professor of Clinical Pediatrics at IU School of Medicine. Dr. Sokol is a clinical neonatologist with particular interest in the treatment of advanced respiratory failure. And he will talk about the impact of COVID-19 on childbirth and the newborn. Please, Dr. Sokol.
Okay. Well, I would like to present some preliminary data on the impact of COVID-19 on childbirth and the newborn. I have a number of disclosures and receive funding from a number of funding sources. However, none of them are related to the work I'm going to discuss today. Well, COVID-19 is a recently identified illness caused by the SARS-CoV-2 virus that results in a spectrum of human illness that ranges from asymptomatic to severe ARDS. It spread into a global pandemic, entering the United States probably in early 2020. From the pathophysiology, limited was known about this clinical illness, especially its impact on pregnant women, childbirth, and their offspring. And we hypothesized the quickest way we could obtain information on the impact of this disease process on pregnant women and childbirth was to establish a real-time national registry for perinatal COVID infections. So to do this, the section of neonatal perinatal medicine of the American Academy of Pediatrics organized to develop a registry. This registry was to be focused on pregnant women and their newborns in the United States. It was a voluntary, real-time, de-identified data collection of pregnant women who were presenting for childbirth with documented SARS-CoV infection, or a mother who was under the investigation for suspected COVID-19. We collected a number of variables on both moms and neonates, and were able to collect or to organize approximately 270 birthing centers that encompass 47 states, including the District of Columbia. And all of these birthing centers account for roughly 18% of U.S. births. Now, the information I'm going to be discussing further is unpublished preliminary data that was dispersed to all the participants in a regular manner through the emerging COVID epidemic here in the United States. The data that is being showed now has been collected and summarized up to September, and I'm showing the data that was shared with all of the co-investigators. So if you look at the scope of the database at this point in time, we had data on 3,722 mother-infant dyads. 98% of these infants were inborn. The demographics, primarily a Caucasian population, 24% Black, 4% Asian. An equal distribution of Hispanic versus non-Hispanic. If we look at mother status at admission, we see that the vast majority of moms, when presenting for delivery, even though they were COVID positive or suspected of COVID, were asymptomatic, 77%. 19.4% did have symptoms. And again, this data collection is based on those who tested positive for COVID-19, which we had data, which the cohort is 90%, and 10% who were mothers who were suspected of COVID-19 infection. If we look at the impact on mode of delivery, we observed that 64% of moms were able to deliver vaginally. 36% ended up delivering via cesarean section. If we look at the birth statistics of their offspring, the median gestational age was 39 weeks. However, The cohort ranges from 15 weeks to 45 weeks. A average birth weight of 3,098 grams, which had a range from 482 to 5,465. Again, this includes the very preterm infants included in the study. And if we look at sex, males were just slightly more present than females. If we look at needs for infant resuscitation, 75% of the newborns did not require any special resuscitation. 10% required some supplemental oxygen. About 13% required some form of positive pressure assistance via CPAP or assisted ventilation. And a small percentage, 2%, required intubation in the delivery room. And remember, this population includes preterm infants. If we look at the neonatal symptoms present, and I would like to focus you on the lower and right side of the slide, the majority of infants were asymptomatic, 86%. If we look at the most common symptom, it is respiratory distress at roughly 14%. Now, if we look at the severity of the respiratory distress, and for that I direct you to the left-hand side of the screen in the upper portion. Again, the majority of infants had no respiratory distress. 3% of the newborns required oxygen supplementation. 8.5% required some end-distending pressure or CPAP. And 3% required assisted ventilation. Now, in the early days of encountering this pandemic, there was a lot of uncertainty over how to house the infants in the hospital. And some centers separated moms and babies. However, if we look at those that were housed with the mother, we see that 57.5% of infants were maintained with their mother, which means that 41.5% were separated from their mother. Some of those required NICU admission, 25%. However, another 17.5% were separated from their mothers and just housed in a different part of the nursery. What type of isolation did these infants have? Well, the most common isolation was contact, droplet, and airborne isolation, which 1/3 of the population received, followed by just a combination of contact and droplet isolation, and then a combination of contact, droplet, airborne, and the baby within a negative pressure room. So COVID infection also impacted the way that babies were fed. If we look at the amount of infants that had direct nursing with their mother, it was 43.4%. Another 8.2% were fed express maternal milk by their mom. Another 15% were fed express maternal milk by someone other than their mother. 3.5% received donor milk, and 30% were formula fed, which is a vast difference versus the conventional amount of formula feeding in our newborns. If we look at infant disposition, for the infants, looking at how many were tested for SARS-CoV-2, roughly 85%, 86% of the cohort. And of those infants, 52 were positive, which accounts to 1.6% of the cohort. Their duration of hospitalization ranged from 5 days, or was a mean of 5 days, ranging from 0 to 135 days. And we see that the vast majority of infants were discharged to home, roughly 98%. There were 0.9% mortality in this patient population. However, none of those were felt to be COVID-related. If I share some local statistics here in central Indiana, we collected data on 127 mother-infant dyads, of which 3 had at least one positive COVID test. And if one looks at this distribution, 2 of the infants had a negative test as well, which would give us a local incidence somewhere between 0.8 to 2.4%. All of these infants were discharged to home within 2 to 3 days, and they were asymptomatic at discharge. 16.5% of these infants were born prematurely. So yes, COVID has had some impact on childbirth and their newborns. However, most newborns are asymptomatic and going through extra procedures such as visualized on this image may be pushing things a little excessive. Thank you.
Thank you very much, Dr. Sokol, for this very interesting presentation on all this data. While I'm waiting for questions from the audience, so I think your last slide, did that just indicate that you can have both combinations, that the mother is tested positive, but the infant is tested negative, as well as that the mother is tested negative and the infant still is maybe tested positive?
No, the entire cohort, 90% of the moms were positive. For all the babies that we admitted into our nursery, we would do 2 tests on them. And it's somewhat concerning to me that a test done within the first 24 hours of life is positive, and a test done at 48 hours of life is negative. So this was trying to sort out, was this perinatally acquired infection versus a postnatal acquired infection. This infant was born to a mom and father that were both COVID positive, and this infant was housed with the family. So it raises concern for me, albeit 48 hours seems a very short period of time for the infant to be exposed postnatally and develop the infection or by a positive test. However, I display it as this because we haven't adjudicated the final results of will these be determined as prenatal infections or postnatal infections.
Okay, great. And I assume, are there plans to follow up those neonates that had been positive for long-term effects?
We are trying to get 1-month follow-up, but this is a large voluntary national cohort. And the limited funding we have was basically just gone into putting together the data center. So we have a large collection of mother-infant dyads. I'm not sure how much follow-up data we'll be able to accumulate because it's unfunded.
I see. Okay. And one last quick question from the audience. How do the infant resuscitation at birth percentages differ from the non-COVID exposed group? This is from Dorothy Teagarden.
I don't have a direct comparison between the two. However, the numbers that I mentioned earlier, roughly 10% of babies will require some resuscitation at birth. So they weren't, the numbers weren't far off from that number.
Okay, well, thank you so much again for this wonderful talk, and we will move on to the next speaker. Our next speaker will be Zhou Taylor, Associate Professor of Human Development and Family Studies at Purdue University. And her research program is focused on examining risk and resilience processes in vulnerable youth and their families. Her talk today is about challenges, well-being, and coping of new mothers during the COVID-19 pandemic.
Are we good? You can see the slides and everything here as well?
Everything good, yes.
Okay. Okay. Hello, everyone. So my talk today is examining challenges, well-being, and coping of single mothers during the COVID-19 pandemic. Now it's not, oh, there we go. Okay. Okay, so we know that, of course, the pandemic has altered everybody's lives, how we work, how we live, how we connect and see one another. However, the impacts of this crisis are not going to be distributed evenly. So we know, for example, there are a number of families who are going to be more impacted, both in terms of disparities by race, but also with gender disparities. Single mothers are an understudied family population who, prior to the pandemic, already had significant vulnerabilities such as high rates of poverty and emotional distress. It's likely that the COVID-19 pandemic is going to exacerbate some of these prior challenges that single mothers experience. So my work today that I'm presenting is preliminary data from the Strengths in Single Mothers project. This was a project that I started in the fall, pre-COVID. Basically, the objective was to examine mothers, single mother families from a strengths-based perspective, as typically single moms are looked at from a deficit focus. So I recruited 200 single mothers across Indiana. They were ranged from their 20s to their late 40s and 50s. Mean age was about 37. This wasn't a very diverse population. It was 88% white, and the mean income was $30,000 to $40,000. In addition to these 200 mothers, 22 of them were participating in qualitative interviews. So the work I'll present today is mixed methods. When COVID hit, I had thought a lot about these mothers, and I had an opportunity to collect data again in the summer, this summer during COVID, and 165 of those mothers came back and completed their surveys, and I conducted interviews with 18 of them. Although, as I said, this wasn't a particularly diverse ethnically sample, there was a lot of diversity in terms of the ages of children, education, and marital status, as you can see here. So on average, mothers had 1.5 children, and you can see their different developmental ages here. So it ranged from moms with young kids to middle school, adolescents, and then a mix of both. Education was quite diverse, which I was happy to see. So almost 20% of the mothers had a graduate degree or above, but we also had a range of mothers who just had a high school education or some college, so that made up almost 50%. And then marital status varied by whether they were divorced and never married, with a smaller group being separated or widowed. So the focus of my talk today is preliminary findings that I'm looking at in terms of a focus on mental health that is strengths-based. So I'm presenting some initial findings looking at gratitude and the effect of gratitude on mental health. Gratitude is a life orientation towards noticing and appreciating the positive in life, and this is often called a strengths of the heart trait, along with other things like joy, optimism, these kinds of dispositional traits. Although these are dispositional, there is a lot of research showing that gratitude can be cultivated. So writing down what you're grateful for, like this graphic here, or writing a letter to someone explaining what you're grateful for. have been found to boost mental health in different populations. However, there isn't any research that I know of to date that has looked at gratitude in single mothers. So my research questions were looking at gratitude, mental health, and stresses pre-COVID, so in the fall in our first wave, on the mental health of single mothers during COVID. And my main focus was looking at how gratitude protected or buffered mothers against declines in mental health. And I'm going to present some preliminary findings that use both quantitative and qualitative data. So this was the main quantitative findings. Essentially, I'm looking at these traits from pre-COVID to COVID. This time period was about sort of 6 or 7 months here. And you can see there's a fair amount of stability in both stresses, internalizing problems, and gratitude. But I was pleased to see that gratitude at time 1 predicted, negatively predicted internalizing problems at time 2, even controlling for prior levels of mental health problems. We do see the effect of stresses, the long-term effect, which is consistent with literature showing the detrimental effect of negative life events on internalizing problems. And the stresses I should note here is negative life events, economic stress, and food insecurity. So moving to my qualitative work, I wanted to pull in some information showing sort of some of the main stresses that the mothers discussed in their interviews and showing some quotes here. So one of the things that they specifically found was that many of them talked about losing support. So these are all pseudonyms here that we can see. Nicole, age 33, she says, one of the hardest things is losing all of the support, so especially the social supports that I think single moms rely so hard on. There is no one to tag me out as a sick mom. Moms also varied in their economic hardship, but we found especially those who were younger and with less education experienced stress resulting from economic hardship, as Amy discusses. My daughter is immune compromised, so I don't want to risk getting her sick, and myself as well. I have really bad asthma. So we haven't had any income in the house within the last 4 months. It's been really difficult. It's been life-changing. Some moms were still working. Mostly we found that it was moms with older kids. This is one mom who was working outside of the home, and she says, I'm away from home. I was working 50 to 60 hours a week just because of the demands of my job. I told the kids, you know, I'm buying food, but you're going to have to fend for yourself. And then homeschooling, which we have been hearing a lot about in terms of all families, and especially the burden falling on mothers here. That this is Mia, age 33, and she said, we as parents all sort of became teachers overnight, and now I'm trying to figure out what I'm supposed to do with my kid during the day while I'm working. And this mother was one of the few mothers we talked to whose ex-husband was involved. So overall, most of these mothers were not being supported either emotionally or financially by their children's father. Okay, so I'm sorry, my slides are not popping in as they should have. So you've seen everything all at once here. So if you look on the left side, this is looking at mental health. So I wanted to sort of see what mental health looked like for these mothers during COVID. So the pie charts there are looking at our quantitative data in terms of depression and anxiety. This was using an NIH measure that had T-scores. So when we look at these are based on these T-scores. So you can see for both depression and anxiety, we had cases of severe levels, especially for anxiety. And for the most part, sort of we see 60% of mothers reporting some form of depression and almost 70% reporting anxiety. This was sort of also shown in the qualitative analyses as well. So I'm just giving you an example of high mental health, medium, and sort of low. So the first one is back to our young mom here, and she says, I'm not doing very well mentally, but I have to be okay for my child. I've been doing the best I can, more anxiety and feeling worthless. Like that's 100% how I feel, anxiety-ridden all day long to the point where it makes me sick. Other people were doing a bit better, but still very aware that their mental health was difficult. So here, sometimes I'm so stressed, I don't react well, tensions are high. I find myself being moody and irritable, and I wish at the beginning I realized I had to take care of my mental health. And then the last person here is doing better. She says she doesn't play this what-if games in terms of thinking about challenges. I'm a survivor, so I typically am resourceful and get through whatever comes my way. So I know I'm running out of time as I thought I might be here, but one of the things that was very dramatic in terms of our interviews, there were a lot of challenges, but unexpectedly a lot of benefits to the pandemic, especially for single moms who I talked to before, who often were very busy. So I have, and I won't go through them all here, but a number of moms basically talked about the benefits of slowing down. So here is an example. And so everything drastically slowed down, and I feel like we are also able to learn to appreciate the downtime in life. And there were some disappointing things, but at the same time, it's like every day is kind of wide open. And I don't remember the last time that life has been that way. So a number of moms really talked about the benefits of being home with their children, even though there was a lot of stress that went along with that. And lastly, we didn't directly ask about gratitude, but a number of people sort of spontaneously talked about gratitude. And this could be sort of thanking that children were old enough, or really feeling blessed for the time that they had with their children. And I'll just give you one example here. This is Mia, and I'm grateful that I do have a child. I have someone who loves me, and just knowing that I do have my child and someone that's going to be hopefully a constant source of love and fulfillment in my life gives me hope for the future. I think if I weren't a mom, I would be bored to tears right now. What would I do? I don't know. It would just be awful. But it's actually one thing I'm really grateful for if you've got to find a silver lining. So we had a number of moms who, throughout their interviews, really kind of pulled in the gratitude piece in terms of when they were discussing the challenges. So implications here, of course, the pandemic is affecting everybody. Single moms are likely to be an especially vulnerable population, and I'm seeing this in these preliminary findings with high levels of mental health problems. Stresses were very high and supports were often very low. So we see a lot that, especially the pandemic, was reducing support from, say, parents that single moms typically relied on. Initial findings are showing gratitude could help buffer single moms against poor mental health. And, you know, I'm looking forward to exploring these findings more in the future. And just thanks and gratitude to the funders. We were funded by a Kinley Trust and the HHS COVID-19 Rapid Response Grant, and my amazing grad students in the lab who I couldn't have done this work without, and my daughter, Suvi, who's given me insight on both the joys and challenges of single parenthood. So thank you.
Well, thank you so much. This is really very touching also to have shared all these comments from these women, and I'm sure that many in the audience can really relate to some of these feelings, even if we're not single parents. Mm-hmm. Do you have any, just a quick question because we are behind on time.
Sorry.
Do you have any insight? I assume that single fathers will have very similar feelings and maybe even more hardship. I just heard a report on the radio yesterday that they find themselves in situations they just have never had before in their lives. Sometimes when fathers now have to stay home alone with the children, do you have any insights on that as well?
I don't have any insights necessarily to dads. Of course, it's a much less common, but, and as I said, there are very few moms who were sharing custody kind of 50-50 in out of the 22 interviews, there was only actually one mom who really had 50-50 in terms of that. So I would imagine that single dads may be less used to having their children sort of to them, but maybe not as dire in terms of the supports, because I think that oftentimes there are single dads that are splitting custody with the moms. Not always, of course. So yeah, but no, it's a good question.
Okay, well then, thank you so much.
Thank you.
Last but not least, I'm excited to introduce Dr. Xiang Zhou, Assistant Professor of Counseling Psychology, who is interested in research at the intersection of culture and parenting. He will be talking about maternal and paternal response comparison at the beginning of the COVID-19 pandemic. So maybe that will give some insight to my question as well. Thank you, Dr. Zhou.
I would like to follow Dr. Taylor to talk about this, because I felt like our data and research sort of complement each other in speaking to different part of parent population. So my name is Xiang Zhou. I'm Assistant Professor of Counseling Psychology. I have my research interests are broadly in ethnic, racial, minority youth well-being, particularly in the context of family. So to present on some preliminary findings I have from a study I conducted during the beginning of the COVID-19, and particularly for the theme of the conference, comparing maternal and paternal responses. We don't necessarily need a lot of research data to know parents are stressed, right? If you're a parent or if you have any friends who are parents, especially of younger age children, you see memes on social media with some of what kind of joy and horrific times parents are having with the children with the lockdown. So similarly with some local policy findings, I have narrative data collected from parents to give you a little bit more context of the project we'll be speaking on. I will be speaking more of later. I collected their narrative data at time 1 of March 31st, so that's about kind of the beginning of the pandemic where we see the rise of cases in the United States. This is a quote from a mother of an 8-year-old son. Says pseudonym Tyler understands well he cannot go to school right now, and like every other child is thrilled with that, which we know is not true for all children, but he can't understand what come over or go to their house. I had trouble making him understand how germs get transmitted. He didn't like what I told him because he was just aggravated and he could not play with his friends and doesn't understand the seriousness of all of this. I felt frustrated because this wasn't our first conversation about it. Like in this quote of how, particularly for younger age children, there are added stressors for parents in terms of communication, in terms of online school learning, in terms of spending even extra time together at home. Of course, positive and negative for some parents. And just last quote I want to share from a father. So again, from a different perspective, perhaps, of a 7-year-old son says, my wife and I were listening to John Prine songs, thinking about his work as he was sick with coronavirus. As asked why we were listening to his music and understood when we explained, he asked if I might die, and we said that it was possible. As pretty commonly has a pretty subdued emotional reaction to specific conversations. It tends to bottle things up and let things out. We've seen a few outbursts from him, but in this conversation, as is typical for him, he just kind of accepted things and went about his day. I wish that I didn't understand as much as he does, but he obviously knows this isn't a normal situation, and we give him honest but simplified answers to his questions about why we're not leaving the house. This particular quote, I think, highlights that the emotional depth in some of these experiences, and what I'm trying to capture in this project is to see how both parents' mental health and children's mental health were co-affecting each other during this special time. Okay. Again, my first wave of data around late. This is, I want to share a little more about the data collection. This is a short-term longitudinal data with 3 time points in the first 3 months of the COVID-19 pandemic from late March to late June. And I highlighted on the timeline of the killing of George Floyd because that may also contribute to a lot of parent health, especially for ethnic racial minority parents. And for today's presentation, I only want to focus on 2 research questions given the time. One is the parental mental health in areas of depression, anxiety. I know Zoe spoke to that a little bit, and also parental burnout. And secondly, in this short time frame, what are the anxiety trajectory look like? A little more about the sample. We collected 495 parents, and only inclusion criteria is have to have at least between the age of 3 to 8-year-old as their target child, and they are currently living in the United States. In our sample, we have 81% mother, 65% white. The mean age is 36, 37 years old. They are from all over the country. It is a convenient sample through parenting agencies and listservs that work before from 41 different U.S. states, with top states being New York, California, and Texas. In my sample, about 20% of the parents are single parents. 52% of parents reported lower annual house lower than 100,000, and 73% reported they were eligible for a stimulus check. Interestingly, between fathers and mothers, we have a higher racial minority parents, fathers. Fathers also younger. We actually have a little more single fathers than mothers proportion-wise, and fathers also reported lower income. So in some of the subsequent analyses, in regression analyses comparing their mental health outcomes, I have called their race, single parent status, annual household, and age to control for those demographic differences of maternal and paternal sample. I want to first present the result on parental burnout. Parental burnout is a relatively new concept in the realm of parenting research. It has received a lot of attention in how they captured parents are feeling emotionally exhausted from their task and role of being a parent that has some overlap but different from anxiety and depression. So sort of tapping into a domain-specific area. Parental burnout assessment has 23 items and into 4 subdomains of exhaustion. That is, one example is I feel completely run down by my role as a parent. Contrast taps into not being the parent they wanted to be and feeling guilty and ashamed of the parent they have become. One sample item here is I don't think I'm the good father or mother that I used to be to my children. Saturation is being fed up with one's parental role, such as I don't enjoy being with my children. And lastly, emotional distancing is when parents feeling they're effectively distancing from the children, such as I do what I'm supposed to do for my children, but nothing more. So across these 4 domains and the total score, total average score of parental burnout, we can see, except from saturation, mothers have higher scores than fathers on all these subdomains after even controlling for race, income, age, and single parent status. On the column, you can see some other significant independent controlling variables. In this case, I think racial minority parents and low-income parents have more parental exhaustion, and younger age parents are even more emotionally saturated. They don't enjoy the time having with their children anymore. But across the board, the gender piece is definitely the most consistent predicting variable in differentiating the group differences. I also want to point out the average mean scores here, given parental burnout assessment new scale. These mean scores are higher than a lot of previously collected clinical samples. Similarly, across the board, a little different. Similarly, we can see mothers either have higher scores of anxiety or depression or similar level as fathers. At T1, T3 in particular, their anxiety scores are higher than fathers. Anxiety and depression here are measured by GAD-7 and PHQ-9, and so these are pretty widely used scales, and there still are a lot of national collected data using GAD-7 and PHQ-9. So one nice feature of using them is we can compare the scores in this convenient sample with more representative national samples, and these are pretty on par with some of the national data that's collected out there. And lastly, this last result slide I want to present is fitting the developmental trajectory or the trajectory of GAD-7 anxiety across 3 months from March 31st to July 1st. You can see on the fitted unconditional growth model here, mother is with a gold color and father with a black color. Using the multi-level modeling, I really want to highlight is fathers started with lower scores than mothers, yet mothers decreased more over time. Another finding I want to highlight is across these times, children's emotional distress at all time points significantly predicted parents' anxiety, which is not that surprising. So the takeaway is parents do experience significant depression, anxiety, and burnout. In a separate study, we have a national representative sample of 2,000 U.S. participants, half of them parents. It's very clear across the board that parents have more stress compared to non-parents. In this sample, we see mothers in general have more depressive, anxiety, burnout symptoms compared to fathers, even after controlling for other important demographic differences. Parents' anxiety from March to June with mothers started higher and decreased more. Not to see an increasing symptom trajectory. We say in the short run, people are relatively resilient in adjusting and bouncing back to their baseline. And lastly, children's adjustment predicted parents' anxiety over time. So hopefully I stay with the time frame.
Yes, wonderful. Thank you. I hope that the next keynote speaker will excuse us to take away some few minutes from him, but are there any questions from the audience for Dr. Zhou? And in the meantime, I was curious about one thing. Does this increased anxiety or depression amongst parents, does that also lead to increased conflicts possibly, or has there been any data on that there is maybe more separations or just conflict situations in general due to feeling more stressed?
Yeah, I cannot speak to that in my data. In the data collection, as you can see, these are actually not dyadic data, so they are fathers and mothers not necessarily from the same family, and there are single parents, so we did not particularly measure conflicts because that might not be applicable to some participant in terms of marital conflicts.
Okay, and the effect on the children to the anxiety of their parents, is that being studied as well?
Yeah, so I'm analyzing that piece across 3 times. I measured both parents' mental health and children's adjustment. We definitely see some mutual influence between parents and children.
Okay, sorry, I misunderstood the children's adjustment then. Wonderful. Very good. Is there any last questions? Otherwise, thank you so much. I would like to thank all 4 speakers again greatly for sharing their exciting research on COVID-19 and for their fabulous talks. I did want to mention to Dr. Sokol that there was still one question in the Q&A box that was directed towards your talk. Maybe you want to answer it in writing there or in the chat. And with that, I will pass on to Dr. Dorothy Teagarden, Director of the Women's Global Health Institute, who will then introduce our second keynote speaker for today. Dorothy.
Thank you, Ulrike. So welcome to our, as she said, our second keynote presentation. And of course, I'm still here to remind you about submitting grants. We have money available. But I also want to emphasize that that is in partnership with the Indiana Clinical Translational Science Institute. And that date is really fast approaching, so watch out for it. But I'm here because, well, I'm very pleased to introduce Dr. Richard Kuhn, who is, and this is a long title, the Trent and Judith Anderson Distinguished Professor in Science in the Department of Biological Sciences, and the founder and Kroenke Family Director of the Purdue Institute of Inflammation, Immunology, and Infectious Diseases, which we all call PI4D. But introducing Dr. Kuhn is particularly gratifying to me because of our very strong partnership with the WGHI with the PI4D, and we're extremely grateful for all the support that the PI4D has given to the WGHI. But his research at Purdue has focused on replication and assembly of the alpha viruses and the flaviviruses. And together with his structural biology colleagues, he has been involved in many fundamental studies examining the structure and assembly of enveloped viruses, including the first structure of the dengue virus, which he's very well known for. His focus continues to be on virus replication, virion assembly, pathogenesis, and host cell interactions. And he's, I think, unique in some ways because he uses biochemical, genetic, and structural techniques. He is the author of over 175 publications and is the editor-in-chief of the Journal of Virology. He's an elected fellow of the American Academy of Microbiology and the American Association of the Advancement of Science. He's the chair of the U.S. panel. I'll be done in a minute, Richard. He's the chair of the panel on the viral diseases of the U.S.-Japan Cooperative Medical Sciences Program at NIAID. And for Purdue, notably, he's one of the Herbert Newby McCoy winners for outstanding research contributions. And not surprisingly for all of us, currently chairs Purdue's Coronavirus Research Task Force. So welcome, Richard. I'm really looking forward to this talk.
Well, thank you, Dorothy. Everybody can see my screen, right?
Yes.
Okay. Well, thank you, Dorothy. And this is a great opportunity to talk with everyone. I think the talks have been fantastic. My talk's going to go in a little bit different direction because I'm going to talk more generally about the virus. I think I'll start with some general comments on the virus. Then I'll dive into, where we are in vaccines and therapeutics, and then I'll finalize with some of the work that other people, other than the speakers that we've already heard, are doing here at Purdue. So, being a structural virologist, I, of course, would like to know the structure of the virus particle. And unfortunately, this is just a model that we have of the virus. We don't have any high-resolution structures of the virus like we have for dengue, as Dorothy indicated, or Zika virus, or many other viruses that Purdue has been known for. There's a number of reasons. The most important reason is that this is a pleomorphic virus and is not easily attacked at high resolution by the tools that we have. But for today, we're really, and this whole pandemic is really relying on the biology of one particular protein. And that protein is a protein that we call the spike glycoprotein, or the S protein. And you can see it, it's this projection off the surface of the virus, and it's shown in red. And there are a number of other proteins that are found in this virus particle, but for the purpose of our talk and for thinking about vaccines, we're not going to talk about them today. You should know that SARS, just like other coronaviruses, has a very large positive sense RNA genome on the interior, and that genome is very large compared to most other viruses in this group, positive strand RNA viruses. The genome is about 30,000 nucleotides, and that offers some interesting opportunities, as we'll see later, in terms of therapeutics. I think we've all heard in nauseating detail things that we need to do to try to prevent the spread of the virus. And clearly, we need to do that as the numbers have been really skyrocketing in every place in the United States. And if you look at the 1918 pandemic flu virus, the amazing thing that people used to say about that is that it really hit every corner of the world. The most remote areas of the world saw influenza in 1918 and 1919. So imagine with our increased transportation that the same is absolutely true here. There is no region of the world that is going to be spared from COVID-19. Now, there are many symptoms, and when you have a pandemic, the numbers of infected people are so large, and we gather this information so quickly that there's a broad range of symptoms. There's the common symptoms, and those common symptoms, in fact, are common for many viruses. But there's lots of additional symptoms given the number of people, the distribution, the heterogeneity of the population. We can see lots of other symptoms. Now, we all know about the severity of COVID-19, but we also have heard about a large fraction of the population, primarily young individuals, but not necessarily, don't seem to get sick at all. And this is not an uncommon phenomena that we see with viruses. And we call it the iceberg principle of pathogenesis. And that is that for many viruses, not every virus, but for many viruses, there is a large fraction of the population that doesn't exhibit obvious symptoms. That's not to say that they're not infected with the virus. That's not to say that they can't transmit the virus. It's just that because of variable factors, including their immune response, they do not show obvious symptoms. There's a smaller cohort, usually, that are slightly unwell, but you wouldn't say that they're sick. Maybe you feel tired. Maybe, you have a little bit of a headache, muscle fatigue. These might be symptoms, but they might not be considered you're sick. And then obviously, once you display obvious symptoms, you're sick. Now, one of the challenging things with a virus that is spread so easily like COVID-19 is that you can transmit the virus when you're in this bottom of the iceberg, that you're infected, you have no symptoms. But you also can transmit virus, and in fact, probably the greatest risk of transmission is right before someone would show symptoms. So even someone who's going to be on this end of the spectrum, before they obviously have symptoms, they can be transmitting high levels of virus. And so given this respiratory nature of the virus, this is a real threat in terms of transmitting to a large number of people. And it's exactly what we have seen with this virus. And it's so frightening because your friends, your parents, your loved ones can appear to be so healthy, and yet they may in fact be sick, and they can be transmitting the virus to you. And that's why we're really stressing all of these precautions to try to prevent transmission of the virus. The virus, as we know, is a respiratory pathogen, and the primary target for respiratory pathogens is going to be in the lungs. And the lungs are rather interesting in that, and especially when we talk about flu, about the kind of seasonal flu versus pandemic flu in terms of the target and what cells get infected. As was mentioned in our first talk, the keynote talk by Dr. Klein, the ACE2 receptor, which is found in lots of different cells, is one that allows the virus to enter cells. And that is found in lots of lung epithelial cells. And probably one of the most interesting aspects of this virus, like a lot of other viruses, is that some of the severe disease that we are seeing is probably not solely the result of the virus itself. It's really the immune system trying to control the infection and the kind of cytokines that are released from immune cells that really trigger some of the severe symptoms that you see. But the lungs are not the only target for this virus. If we're talking about seasonal flu, lungs are usually, upper respiratory tract lung is usually the primary target. You don't usually see lots of systemic spread. In COVID-19, that is not true, in that you do have lots of organs and tissues that are being infected with virus. The heart is one of the primary ones that people have seen, and it's apparently leading to lots of heart problems in people who have even recovered from a COVID-19 infection. So this whole system of tissues and organs that are being infected, I think, is a really interesting biology of the virus. One question is asked frequently is, why didn't we see SARS-CoV-2 coming and prepare ahead of time? And there's lots of responses that we can give to that. There has been a lot of talk about a pandemic coming, whether it's a flu pandemic or whether it's a pandemic like Zika that broke out a few years ago, or whether it's a coronavirus pandemic. And we've known now in the last 20 years that coronaviruses are really a significant threat for a potential pandemic. The first significant human coronavirus was SARS, severe acute respiratory syndrome virus. And that virus was tracked back to horseshoe bats, probably transmitted to these palm civet cats, and somewhere along the way it was transmitted to humans, and human-to-human transmission occurred. And so once you make this jump across animal species to a human, and the human can transmit to another human, you now have a potential for a pandemic, especially in the case of a population of humans who have never seen this virus before. So there's also, that was back, I think, in 2003, and then there was in 2012 another coronavirus that was related to the original SARS virus, which was called Middle Eastern Respiratory Syndrome virus, and this also probably originated and transmitted by bats and went into camels, and then from there was transmitted to humans, and a substantial number of humans are actually seropositive, and so are the camels. So the transmission cycle of COVID-2 is probably quite similar. The origin is in bats, and we don't have time to talk about it, but bats are a frequent reservoir for a large number of viruses that infrequently enter the human population. Usually they don't enter directly from bat to human. There's usually an animal or animals that act as transmission vectors. And we're not sure of the transmission vector for COVID-2 yet. People have suggested this pangolin animal as a possible intermediary, but we don't know. But once it entered the human population, it was easily accessible on the ACE2 receptor, and so it was then able to transmit from human to human. And we now know that it's fairly easy, since it's a respiratory pathogen, to move from human to human through droplets and probably aerosols. And certainly there can be environmental contamination that can also result in transmission. So as you're listening to this talk, please don't put your hands on your face, which most of us do at least 20 to 30 times every hour. But talking about, what is the potential and why didn't we see COVID-19 coming previously? Well, it turns out that we've now found lots of coronaviruses. There are a lot of these coronaviruses that exist in bats. There are some that are in humans, as we know. Prior to the original SARS, we knew about significant coronavirus pathogens, but they were primarily pathogens of animals. Infectious bronchitis virus is a coronavirus that infects birds and is significant in the turkey population in the world. There are also ones that infect cats and some significant ones that have infected pigs. But prior to the identification of SARS in 2003, the human coronaviruses that we knew about didn't cause much significant disease. And unfortunately, usually money follows disease, and usually human disease. And so there was a number of groups that had been looking at coronaviruses and studying coronaviruses. But once there was a human virus that caused severe disease, the number of groups that were studying coronaviruses expanded greatly. And shortly after the 2003 outbreak of SARS, people were able to identify additional coronaviruses that are infecting humans. And it turns out that probably 20 to 40% of human colds are actually caused by coronaviruses. As I said, in 2012, the outbreak of the MERS in Saudi Arabia, and now in 2019, the outbreak of SARS-2. I think we can count on there being future outbreaks. The question is, are we going to be better prepared for it than we were this time around? I should say that we weren't unprepared completely. We moved vaccines and we moved therapeutics, with speed that we've never seen before. And so there is hope in terms of the vaccines and therapeutics. But still, the tools that were available in 1918 for the pandemic flu are still very valuable tools as we begin to mitigate the transmission of COVID-19. So let me just step back for a second. My guess is that you all know this, but I just want to put it in a context. What is a vaccine? Well, a vaccine is a substance that's used to elicit immunity and protect an individual from disease. This is not to say that a vaccine is going to prevent being infected. You still will be infected. It's just that the vaccine will prime your immune system so that you very rapidly can recognize this pathogen that you've been immunized against and react to it very quickly so that it doesn't spread in your body, so that it doesn't cause disease. So that's an important aspect. There is, to my knowledge, there's very little in terms of vaccines that actually prevent an infection. Then we can ask the question, are all vaccines equal? Well, there's lots of different types of vaccines, and we really have to understand something about the pathogen to make sure that we have the right vaccine for that pathogen. And that's why we're still somewhat unsure as to how effective, at least in the long term, the current crop of COVID-19 vaccines are going to be. So where is this COVID-19 vaccine? We've heard lots in the last couple of weeks about COVID-19 vaccines. We all know that there are hundreds of vaccines that are being developed worldwide. Many of these have now moved into the phase 1 safety trial phase, the phase 2 phase of a clinical trial that tries to establish whether these will produce an immune response. And then finally, in phase 3, not only are we looking at the safety and the immune response that's produced, but we're looking to see whether we will have protection and protection against disease, which is the real important point we're after right now. On the left side in pink and purple are the so-called Operation Warp Speed finalists. Those are ones that are being promoted by the United States government, but there are other vaccines out there. I think one important thing to note, and that's the point that I alluded to a few minutes ago, is that we're going to have first-generation vaccines, and we're going to all probably try to take them. There may be second- and third-generation vaccines that may be better, that may provide better protection, or that may provide protection for a longer period of time. But we're trying to do this as fast as possible to make sure that that number of worldwide infections doesn't pass 1 billion or 2 billion people. And this is just looking at the amount of investment that's been made in quite a number of these are hitting the $1.5 to $2 billion mark. A large fraction of that money that's being invested is actually for the large-scale production of these vaccines. And here's the clinical trials that are going on that people are looking at. You know, we just heard, I think earlier, maybe last week, I don't even remember what today is. Yeah, maybe this week, earlier this week from Pfizer, their phase 3 clinical trial, they released preliminary results. In their phase 3 trial, they have 43,000 volunteers that have enrolled. Some of them are receiving a placebo, and some of them are receiving, in the case of Pfizer, as well as in Moderna, they're getting a new revolutionary type vaccine, which is a messenger RNA vaccine. That RNA is essentially incorporated into cells, and those cells then synthesize the S or surface protein of SARS-CoV-2. And in so doing, you will then recognize that as foreign. You'll mount an antibody response against it. And in this case, for this particular sets of vaccines, there will also be a T-cell response against these vaccines, which may be important for long-term immunity. But of these 43,000 volunteers that are moving along the pipeline, the phase 3 pipeline, there's been 94 cases of COVID-19. And really, just to be clear, in order to evaluate how good the vaccine is, we have to have a significant fraction of those 43,000 or 43,000 people. They have to be exposed to virus because we have to be able to establish whether some fraction of them are protected. And in this case, less than 10% of the people who did receive a vaccination were showing symptoms of COVID-19. That's where the 90% value comes in. They would like to have a total of at least 164 cases of COVID-19 infection, to the point where they can then, with some level of confidence, and the oversight committees that review this data say that the vaccine is effective and can be released. As of November 8th, which was on Sunday, they had 89% enrollment. 89% of their 43,000 had gotten a second dose of the vaccine. Before they go to the FDA for an emergency use authorization, they would like to wait 2 months after all of the enrollees get that second dose of vaccine. We expect the Moderna vaccine, which is very similar because it's also a messenger RNA encoding the S protein. We expect them to release preliminary data and probably final data by the end of this calendar year. And then there's a number of other vaccine candidates that are also moving along, such as the AstraZeneca Oxford one and the Johnson & Johnson one, and then there's another one, Novavax. And I'm not going to go into the details of what each of those vaccines are. But given what I just said, would you get vaccinated? I'm not sure what the current number is. I hope that they continue to look at this because it's very important. But some preliminary values that I've heard is that maybe only 50 or 55, 60% of the population of the U.S. will take a COVID-19 vaccine. And then the next question is, why would you get vaccinated? Well, if you're in a risk group, that's a good reason for thinking about getting vaccinated. But what about if you're a young child? And maybe this is not your decision to make if you're under 18, maybe it's your parents. But why would you get vaccinated if the likelihood of severe disease is very low? Well, it's really the kind of the realm of what we call public health. And public health is not specifically interested in individuals, but it's interested in protecting populations. And as many of you have heard, what we would really like to do is have a large fraction of the U.S. and the world population to be immune from COVID-19, because as that number of immune positive individuals gets larger, then the ability of the virus to move from person to person begins decreasing. And we're going to hit a certain point, whether that's 80% of the population or 85% of the population who have immunity. Once that herd immunity, as it's been called, hits the right number, and transmission will drop. And even those people who have not been vaccinated, a small percentage, we hope, will be protected. But let me be clear that we all really should try to be vaccinated, assuming that there is a safe and efficacious vaccine. But let me tell you about, there are quite a number of people who don't want their kids to be vaccinated. And I'm not sure what, there we go. It turns out that those people tend to be wealthier on average, with average incomes more than 4 times the poverty level. They tend to be non-Hispanic whites. They tend to be married, English-speaking couples. And they also usually have a college degree and are fairly well educated. So it turns out that the more educated you are, the more likely you are not to have your children vaccinated, which is a stunning observation. You can see from this graph, which is provided by the CDC, that unvaccinated children, 82% of them tend to be white, with 9% Black, 7% Hispanic, and 2% Asians. That's something to think about. If you ask which country in the world is most likely to have their kids vaccinated, the answer is Bangladesh. So the countries that are most devastated by infectious disease are the ones that are most likely to be vaccinated. And countries like the United States and France, in which infectious disease has been mitigated in large part because we have vaccinated, they're more likely not to vaccinate their kids. One point I just want to raise as we're moving through this is that for quite a bit of time, probably on the order of 6 months, current vaccinations have really been slowed down, if not stopped. And things that are dear to my heart, such as poliovirus, we're on the edge of eradicating poliovirus from the planet, and yet we had to take a break because of the COVID pandemic. And so we've been dealt a setback. We were extremely close. There's only a couple of countries in the world that currently have wild-type poliovirus circulating, and now we've kind of stepped back, and that has significant effects. So this is something that I think we're only going to see in the next year or so, that infectious disease, think about measles, is probably going to be rising again as we kind of delayed or didn't give our children the vaccines that we have available. Now, we're all kind of torn by what to do about this and what role does the government play in this. The government has a role in making sure that we know what's out there, that we are prepared to deal with this, both in terms of developing vaccines, but perhaps more importantly, to have the appropriate protective equipment so that we don't spread something like a respiratory pathogen. We should get clear guidance from the government on how to protect ourselves and our families. What can you do to eliminate infectious disease? Well, I think it's really important that everybody learns a little bit about what this disease is and how it's spread and what it can do. We should be able, once we do that, to change our behavior, to mitigate, to stop, prevent the rapid spread of this pathogen. Hopefully, we're going to have a vaccination, and we will have treatments shortly. And I just want to emphasize that the transmission and the numbers in Indiana have been skyrocketing, just like all across the United States, is that everybody plays a role in slowing the spread of this virus. So I'm going to begin talking a little bit about some of the things that are just going on at Purdue in the remaining 10 minutes or so. My colleague, Andy Messakar, who is in structural biology and the head of biochemistry, Department of Biochemistry, as well as Arun Ghosh, who is in the Department of Chemistry, have been working for nearly 20 years on protease inhibitors against viruses, and particularly against the coronaviruses. And what Andy and Arun have been focusing on have been two of the proteases of the virus. And these proteases are important. It turns out that an RNA virus like SARS-CoV-2 makes a very long protein, a polyprotein that has to be processed by a virus-encoded or virus-encoded proteases. And these proteases are susceptible. They're similar to host proteases, but you can actually design them so that they're susceptible to specific small molecule inhibitors. And although they've been working on this for many years now, in response to the COVID-19 outbreak, they started very early in the pandemic. And since February of 2020, they've synthesized more than 320 compounds that are targeting these two proteases that we find in CoV-2. And some of these have pretty good potency, 1 to 5 nanomolar, and they have also been looking at whether they synergize with some of the other compounds out there, specifically remdesivir, that has been given emergency use authorization. So as I said, these guys have been working since 2003 and have made lots of different molecules. The good thing is that CoV-2 proteases look very similar to the original SARS proteases, and so they really had kind of a head start. Should point out that Professor Ghosh was the person who invented this compound called darunavir, which is in the clinic against HIV. And Andy and Arun have done multiple patents around this. One of that is for this molecule that they call X77. I'm not going to go into the technical details of this, but the beauty of the Messakar-Ghosh collaboration is that Professor Ghosh is a medicinal chemist who knows how to tinker with molecules like this one right here, and Andy Messakar is a biochemist and a structural biologist who knows how to do structures of the protease in complex with these inhibitors, knowing that you can now tweak it to increase the activity and you can increase the bioavailability and other properties of these small molecule compounds to make them more and more drug-like. Andy Messakar and myself are the Purdue representatives for an NIH and NIAID funded contract for solving structures of proteins from infectious agents, both viruses and bacterial pathogens and even fungal pathogens. And the center has done, actually it's far more than 34 structures now of 12 unique proteins. But there's every day there are more structures that are now coming out from the center and in other places all around the world. So in the last few minutes, I'm just going to walk through very quickly the programs that we have at the Purdue Institute of Inflammation, Immunology, and Infectious Disease, PI4D, as Dorothy noted before. We have a number of program areas. We have more than 200 faculty that are affiliated with it. And early in this pandemic, we got together whatever faculty were interested in. And so we've been trying to coordinate some of the opportunities and some of the players in that. And this is just the COVID research task force that we put together that Dorothy mentioned earlier. So I'm just walking through, there's a set of several faculty that are developing new technologies for COVID-19 detection. I can't stress enough that, yes, we do have some testing capabilities, but I'm sure you've heard in the last few days that those testing capabilities are running out right now. What we would really like to have is point of care detection so that you don't have to go too far to be tested. And Jackie Linus, who is a professor in the Department of Biomedical Engineering, has been working on this lateral flow technology that allows for rapid detection. She's also done against a number of other pathogens, including HIV, which is deployed in Africa right now. Professor Mohit Verma in the Department of Agricultural and Biological Engineering has also developed a test for COVID-19. This is a saliva-based test, and they are working with the company Raytheon to actually produce this and get it out on the market. And then finally, in the detection space, Leah Stancha in the Department of Materials Engineering has a completely different technology for the detection of SARS-CoV-2, and it looks at differences in conductance of electrical signals, looking at bound and unbound RNA from SARS-CoV-2 virus. And this partnership that was started is with a company called Identify Sensors, and Leah and her colleagues have started a spin-off of that company to further develop this sensor, this biosensor that they are developing. I've been working with a Purdue alum and a good friend of Purdue named Doug Campen with some material for disinfecting surfaces, and we have been testing his compounds out in our BSL-3 facility. Some of our staff that are working in the BSL-3. This is a lab in which we actually can work with live virus, and we have substantial amounts of virus. And I should also say that we're working with a number of groups, especially with Andy Methkar and Arun Ghosh, to do live virus experiments, testing their compounds for inhibition of the virus. And there's Andy Methkar explaining things much better than I can. Yun Yao in the Department of Food Science is repurposing like niclosamide as a therapeutic. It's been shown clearly in the case of SARS that it inhibits virus replication, and it's currently in clinical trials to determine its effect on COVID-19. And then kind of in a different direction, Andrew Welton and Caitlin Proctor in the Department of Civil Engineering are looking at the effects of buildings that have had their water shut down for a long period of time and how that influences water quality. With the spectrum of faculty, we heard some of them speak earlier, the spectrum of faculty that we have at Purdue, there's a large number of faculty that have turned their attention and their passions toward looking at ways that we can mitigate this virus. And I will just end with Rob Stalin. He's in the Department of Medicinal Chemistry and Molecular Pharmacology. Rob is a virologist who uses biophysical tools to understand the membrane interactions of these viruses. And SARS-CoV-2 is a membrane-containing virus. This is the life cycle of the virus. I'm not going to go into any details, but Dr. Stalin is really interested in the step here in which the virus is forming a particle itself, and it's acquiring a lipid envelope and how it moves in and out from there. He's expressed a number of proteins. Those proteins you can put together to form what looks like a COVID-19 virus, but is not infectious. We call these virus-like particles. And he's been able to show that these virus-like particles require specific lipid molecules, and therefore this is a potential therapeutic, although it's a little bit further downstream than the Mesicar-Ghosh compounds. And so with that, let me just end by thanking the Women's Global Health Institute. Make no mistake, COVID-19 has impacted women in many significant ways, and we're probably not going to know all of the ways that they have been impacted. And so drawing attention to it by bringing people who have a passion for this together, I think is really important for studying and understanding the differential impact of this pandemic on women. So with that, thank you.
Thank you, Richard. I really appreciate it. It was so interesting because I think it addressed a lot of things that we are all very interested in and thinking about. I think we're open. I'm sure we'll be happy to answer any questions. So if you have a question, please be sure to enter it into the Q&A box. In the meantime, while waiting, what's the probability of a COVID-19 mutation that could partially or totally invalidate a vaccine? What do we know about it that might happen?
Yeah, great question, because there are reports that these are viruses that do change rather rapidly. The bottom line is the way the vaccine is developed will probably have it will elicit multiple antibodies. And so hopefully we're not going to be dependent upon one specific antibody that the vaccine will elicit. We'll have a collection of them. And so if you change in one site, you might lose one of the antibodies that is generated, but you'll have other ones. But this is one reason why I mentioned that we need to be thinking, and we are thinking, about the second and third generation of vaccines.
Okay, great. I have another question from Belinda Cito, who's one of our External Advisory Committee members. Richard, great talk. Would you describe the data platform for the center? How are data shared?
Hi, Belinda. Good to talk with you. I would say that the data platform has been bits and pieces. So we've been working with a number of entities in the state on data sharing, but I wouldn't say we have a kind of connected data platform across Purdue. Not having the medical school right here, I think limits some of the things that we might do in terms of patient data.
Yes, but it seems like there could be ways, and I guess this comes to another question I have is, how can we position ourselves, Purdue, to do the best research and rapid response to infectious diseases in the future? How can we coordinate our responses well on Purdue's campus? And I think your work in the PI4D has been one of the best ways we have, but do you have any suggestions for all of us for how we move forward in the future?
Well, I think, Dorothy, as you know, Tommy Soares has been trying to coordinate as much as possible. And so for those groups that want data or have data, Tommy will coordinate both internally at Purdue with the folks in the Data Science Institute, as well as people, for example, at the Indiana Biosciences Research Institute, because they also have some data platforms that they've built up for other purposes, but are now being kind of integrated with COVID-19 data. So if any of you have a need for data, or you have data that you're not sure how you can assimilate it with other data, Tommy Soares is the guy to talk to, and his email is very easily found on the Pi4D website.
Thank you, Richard. I think that's our last question. So I just want to thank you again really a lot for this presentation. It was really helpful to hear it, to hear the Purdue's perspective. So thank you.
Thanks, Dorothy.
We look forward, I'm going to harp on this again, to pilot grants. Be sure to look for it on our website. If you're interested in the WGHI future events, which next semester we'll have another group of events, please watch on the WGHI website, and you'll also find the recording of the symposium posted in a few days, probably, as soon as we can get it up on the WGHI website if you're interested. So thank you all for participating, and thank you again to all of our wonderful presenters. Have a wonderful afternoon and weekend.